I'm here to welcome you all to the 2016 Lynford Lecture. I'm especially pleased to welcome the lecturer, Professor Bonnie Bassler of Princeton University. Jeff Lynford will give a proper introduction to her later on. So I will move on and say a few words about the lecture itself and about Jeff and Tondra. If I don't say it, nobody else will. This lecture is the 17th in the series. I've often heard people say if they do things more than twice, they'll call it a tradition, but 17 is probably qualifying for the word tradition, and it's a very important one for our school, something to which we look forward to every year. It would not happen without the support and active involvement of Jeff Lynford. My wife and I have come to know Tondra and Jeff Lynford better with each passing year, and I have begun to appreciate the goodwill for the enormous number of causes they espouse, among them NYU and our school. For instance, Jeff is a member of the NYU Board of Trustees and also a member of the Board of Overseers of our school. In my view, the Lynfords represent the best of the American spirit of giving to others whenever possible. I appreciate this a lot. Jeff will soon be on the stage and make his presence felt, but Tondra, on the other hand, will not come on the stage. So, Tondra, could I please ask you to stand up and wave your hand to the others? Thank you so much. There is another important part of the Lynford Lecture. The lecture will be followed by a discussion of the lecture itself by a panel which consists of the lecturer herself and Dr. Martin Blazer. I saw him earlier, there he is, and Professor Daniel Cronauer of the Rockefeller University. There. Welcome to both of you. To you as well, Jeff will provide a proper introduction, so I will not step on his toes. Finally, there is a committee that chooses the lecturer each year. The two people who have been steadfast members of this committee are David Chudnovsky, who is here, and Gregory Chudnovsky, who is sitting here. They bring their wisdom to bear on the selection of the fine lecturers such as today's. And Jeff, of course, hovers around this with a benign presence. Thank you, gentlemen, for this. I am also a transient member of this committee, so I deserve a little bit of credit as well. But I shouldn't overplay that. So. The lecture series has had very distinguished people in the past. If the American electorate so chooses, one of the past lecturers may well become the next president, and I'm not talking about Mr. Trump. Ladies and gentlemen, welcome to this lecture. Jeff, now you can come here and dazzle everybody with your remarks.
President Srinivasan, fellow trustees, Professors Chudnovsky, faculty, students, and honored guests, Dr. Bonnie Bassler of Princeton, Dr. Martin Blazer of NYU, Dr. Daniel Cronauer of the Rockefeller University, and their spouses, Mr. Ellis Rubenstein, President of the New York Academy of Sciences, and Mr. Pat Foy, sitting way back there, wave Patrick, the Executive Director of the New York and New Jersey Port Authority, or the Port Authority of New York and New Jersey, where I spend some of my extracurricular time. This afternoon, my wife Tondra and I are happy once again to participate in this annual lecture series. It began in this auditorium in 1998. The brothers Chudnovskys, founders of the Institute for Mathematics and Advanced Supercomputing, were the co-founders. They were actually the force behind it all. And we've been able to attract, through their hard work, a variety of illustrious speakers. We've had 3 Nobel laureates, 2 Alan Turing Award winners, 1 Fields medalist. And today we're happy to have what I believe is our first MacArthur Fellow. Dr. Bassler. Where are you, Doctor, again? Okay, she's hiding. Okay, and our second female lecturer. And when I told that to her, I was chastised roundly for having only two, and you're right. But you have to stand behind my wife, who has been after me for 17 years. And so you're making me look good today, and I thank you. We've had one other MacArthur Fellow, though. Sitting to your left is Gregory. And any other MacArthur Fellows in the audience that I've somehow skipped? Who would like to be a MacArthur Fellow? Okay. To start, I would like to quote from the noted author Stephen Jay Gould. We live in an age of bacteria. I'll let that resonate. This is because they've been on the planet approximately 3.5 billion years, and humans have only been here 200,000 years, and civilization as we know it, maybe 6,000 years. So we have asked Professor Bassler, the noted researcher in the field of molecular biology, to talk to us about why is this? How did this happen? What does it mean for us now and in the future? And so her talk, entitled Tiny Conspiracies, Cell-to-Cell Communication in Bacteria, starts to unravel that puzzle as to how they have been able to last so long and what that means for our modest existence here. She has been the author of the concept, or the popularization of the concept of quorum sensing. Now, did you invent that term? You made it better. Okay. So quorum sensing is one of three words that are three concepts that this is your homework. Before you leave tonight, you'll have to figure out what is the intersection of quorum sensing, epigenetics, and microbiome. Each one of our speakers is an expert in those areas, and hopefully they do intersect, and by the time you leave, you'll have some feel for that. So, I accidentally pushed your slide, I'm sorry. One of the reasons we asked Professor Bassler, may I call you Bonnie, to come is that we at NYU are trying to change from the historic chemical engineering, electrical engineering, mechanical engineering, and broaden our scope into biomedical engineering and other forms of the melding of biology and medicine. And for example, we have the Othmer Jacobs Department of Chemical and Biomolecular Engineering, where we offer programs to employ current trends utilizing novel molecules, advanced products and delivery processes, as well as synthesis, design, and operation methodologies. That's a very complicated way of saying what they invent at NYU Langone, they need us to figure out how to deliver. And I think that's going to be very, very important for NYU Tandon. I think I'd like to conclude, it's a little backwards, talking a little bit about Professor Bassler's academic and professional achievements, because I know that many students ask the question, well, how did he or she get where they are? What was their secret? Do they have a secret? What did they study? What was the career path? And so I'm going to give you the career path of each one of our people here today. But Professor Bassler got Phi Beta Kappa degree in biochemistry from the University of California. She then got her PhD in biochemistry at Johns Hopkins. And in 1994, she joined Princeton University, which I had graduated a quarter of a century before, and she's been there ever since. So you must be doing something right. In 2002, she received the MacArthur Fellowship. 2005, she became a medical institute investigator for Howard Hughes Institute, which is a big deal. Does anybody remember who Howard Hughes was? He was one of the wealthiest men in the world, and he set up a medical institute. She also was elected to the National Academy of Sciences. She's a fellow of the American Academy of Arts and Sciences, and she was appointed by President Obama to the National Science Board, which oversees the National Science Foundation. So she has clearly spread her talent among many fields, and in addition to her 135 research papers, her 3 honorary degrees from Swarthmore, Bates, and Tufts, and the 24 patents that have been either issued or pending. She has been a role model for female students to become scientists and engineers. And these are fields that have historically not been fertile enough to see the importance of female scientists and engineers. And so she has been encouraging institutions and organizations to try to rectify that. And so without further ado, Professor, please come up. And you're going to give the award, so why don't you come up and give her the plaque.
Alien communications occupies us every day. We learn about some unusual planets out there which give signals or whatever. And of course, communication is a very important thing, whatever it is, because it allows you to change the structure of objects which communicate. Okay. Communications are also very important for another reason. Without Alan Turing and efforts of people in Bletchley who were able to decipher the Nazi communication, it's not clear what would have happened. However, we have to invert this thing, and we have to be aware that instead of looking up there, we should look actually inside. Because depending on how you look at it, but it's a lower bound. For every cell of our body, there is at least one bacterial cell in us, which communicates and does some other interesting things. So this is where the research of our speaker comes in. She allows us to look on this alien communication system inside us and harness it for the benefit of the humankind. And this is very important thing. And this is why here is the plaque, and it's my pleasure to present it to our speaker.
Tondra, look up here, picture taken. Trini, you want your picture taken? Come on, you're the president of this place. You're allowed to have your
He's thinking about it.
Yeah, right. Thank you.
Thank you. Well, thank you. I'm just enormously thrilled to be here and humbled to receive this nice award. It's extremely meaningful to me as your neighbor and an admirer of this institution. I want to thank the Lynfords for having me and hosting me, and I want to thank the Chudnovsky brothers for finding me a few years ago. Anyway, I'm really, really grateful because the best part is I get to meet all of you. So thanks for having me. And I also want to tell you some science, right? I got a plaque. You should figure out why. And so anyway, I want to just give a short seminar on the work that my gang does at Princeton, right? Which will sort of set the theme of what we're going to talk about in a little while. So you've actually had a really wonderful introduction twice to my work. And so what I work on in my lab is how bacteria communicate, how they count, and how they carry out group behaviors. And then what we'd like to do is to be able to manipulate those conversations to make good bacteria better or to stop harmful bacteria from doing things that make us sick. And so that's what I'm going to talk about tonight. And so to get there, I want to make sure that everybody knows what bacteria are. And so I'm just going to start with a simple cartoon of the critters that are at the heart of my studies. So you already heard that bacteria are the most ancient living creatures on Earth. They've been on Earth for a few billion years. They all look pretty much like this guy. They are single cells, and you need a microscope to see them. They're covered in a membrane, which is like their skin, and so that keeps the outside out and the inside in. And inside of bacteria are all the biomolecules that keep them alive, and then also in here in blue is the DNA, which is the genetic blueprint that has the information for bacteria to make more of themselves. And so if you just watch a bacterium under a microscope, they're pretty boring. What they do is they consume nutrients, they double all of these components, they grow to twice their size, they cut themselves down the middle, and one cell becomes 2, and 2 becomes 4, and so on and so on. And so they eat and divide and eat and divide. And so scientists have known that for hundreds of years, since bacteria were discovered. But the question for me, or what they've always thought about these bacteria, is because they're so simple looking and they kind of live this simple life, that they were actually asocial recluses. And what I mean by that is when they divide into 2 cells, the sister cells have no knowledge of their sibling. But now, when we know about bacteria and we think about all of the things they do, the question that my lab had was not that they can divide in half, but how do they get any bang for their buck? So historically, you've heard about bacteria because they cause disease. And so these are just some pictures of famous bacteria taken under a microscope that cause diseases that you've heard of. Increasingly now, we're going to hear about this magic thing called the microbiome. That's one of the 3 words. I always give the test answers away at school too. So anyway, we're increasingly learning that bacteria do all these wonderful things that are essential for life. So as you heard, we live in a consortium with all of these bacteria, particularly in our gut. And what they do is all kinds of processes that your human body can't do. So for example, they digest plant food. If you eat a salad and they give you access to those calories, they educate your immune system to keep harmful microbes out. They make lots of our micronutrients, all kinds of big jobs that keep higher organisms alive. And so the question that is at the heart of my lab's work is whether you want to think about all these bad things, like how do bacteria, these itty-bitty critters, kill us, or you want to think about all these good things, how do these itty-bitty critters keep us alive? Our question is, how can they do either of those things? They're so small. How can it be that they have this enormous impact on this earth? And so what I want to try to show you is the way bacteria do it is by acting in groups and carrying out jobs as collectives that if they acted alone, those jobs would be inefficient. And so the way that we got to that understanding that these ancient bacteria could act in groups actually came from a kind of funny observation. It started with this animal, which is the Hawaiian bobtail squid, which lives in a beautiful one-to-one symbiosis with a bioluminescent bacterium named Vibrio fischeri. So Vibrio fischeri lives in a specialized light organ of this squid, and it gives the animal light. And so now I want to show you, talk to you about this symbiosis. And first I'm going to tell you from the squid's perspective. So this animal, it's about this big, full grown, and it's nocturnal. So during the day, it sleeps buried in the sand, but then at night, it has to come out to hunt. And so what we've done in this movie is we've put the squid, it's in an aquarium, and what we've done is we've taken away the sediments and the light is on. And so what you can see is it looks kind of spastic, and that's because it's never meant to be out in the light. If it was out during the day, a predator would see it and would eat it. So now what we've done is we've given the animal sediments, the light is on, so it thinks it's morning, and it thinks it's supposed to go to sleep. And of course, it has to stay away from predators during the day, too. So you just saw it camouflage itself. And so now what you see is that it's burying itself in the sand, right? Because you only get eaten by a predator once, right? And so it has this fantastic strategy, right, during the day to keep away from predators. It just looks like the sand. It buries itself just a couple of millimeters under the top layer of the sediments. And if I showed you this aquarium, you wouldn't know there was a squid in there. There's actually about 10 of them. Okay, so this is a fantastic strategy during the day to stay away from predators. But still, this animal has this problem because at night it has to come out to hunt. It has to eat, like I told you. And so it's called the Hawaiian bobtail squid because it lives just off the coast of California, but only in knee-deep water. And so when it emerges at night to go hunting, since it's in shallow water, on bright nights when there's lots of stars or moonlight, that light can penetrate the depth of the water. And if the squid was swimming around and the starlight shone on it, it would catch cast a shadow, and then predators that see its shadow could find it and eat it. So it's got to have a way to stay invisible at night, and this isn't the way. And so that's where this bacterium comes in. So now what you're looking at is the squid has been turned on its back, and what I hope you can see, it's a little bright in here, but there's two glowing lobes. Inside that is a specialized light organ that houses this bacterium, Vibrio fischeri, that I told you is bioluminescent. And so so it makes light, like fireflies make light. And so what happens is when the squid comes out at night, it's got this light organ filled with this glowing bacterium. And then the squid has sensors on its back, so it senses how much starlight or moonlight hits its back, and then it opens and closes a shutter. So the amount of light coming out of the bottom, which is made by the bacterium, exactly matches how much light hits the squid's back. So when the squid swims, it doesn't make a shadow, because it counter-illuminates itself with the light made by the bacteria. So the anti-predation device at night requires this partnership with this beautiful glow-in-the-dark bacteria. And so that's how it works. And the reason the bacterium is willing to provide the light is because it can grow to really high numbers in this light organ. So the squid puts all kinds of food and goodies that the bacteria can live on in this light organ. And so it's a lot easier to live in the light organ and make more of yourself than out in the ocean, which is kind of a nutrient desert. Okay, so now you understand that both the bacterium makes light because it gets fed, and the squid uses the light, right? And this symbiosis has evolved over time because they each get something out of it. But what's really interesting from my point of view is about the bacterium. It only makes light when it's inside of that squid. And so what we wondered is, how does the bacterium know whether it's outside or whether it's inside the squid? And so this has to do with the bacteria talking to each other. So now I'm going to tell you how the bacterium knows to turn on light. So this is supposed to be a cartoon of a bacterial cell. And so you have to remember, these cells are small. So the world is big and the bacteria are small. So if they're out in the ocean, they're in the big world. And so they're at low cell density, right? And so what happens is that when these bacteria are at low cell density, they want to have the program of gene expression going that's good for acting as an individual. So they're doing some subset of the tasks that they're doing, not making light, but among the things they do is that they make and release small molecules that I have with these triangles, these red triangles, and we call them autoinducers, but you can think of them like hormones. And so what happens is when the bacteria are at low number, they release these autoinducers, the autoinducers float away, the bacteria can't detect them, and that says you're an individual, you're not in a group in this squid, so it means for Vibrio fischeri, don't make light. But then, as the bacteria grow and divide, since all of the cells are making a share of this hormone, this autoinducer molecule, the concentration of this molecule, depicted by these red triangles, outside of the cell increases with cell number. So the more cells there are, the more of this molecule there is. And when the molecule hits a particular amount, the bacteria detect it, and what they infer from that detection event is that they must have neighbors around, because only at high cell density could that molecule accumulate. So in unison, all of the bacteria detect the molecule and they turn on light together. And this situation only happens in the light organ of the squid. So the bacterium doesn't actually know it's outside or inside, it's actually measuring cell density, and the high cell density condition is in the squid. And we know this is true because we can just centrifuge the cells out and collect the liquids, which has that molecule in it. And if we squirt those liquids onto these cells, they'll turn on light, right? So they don't know how many cells there are. They're just measuring the chemical, and they use it as a proxy for cell number, right? So then we wanted to understand, how does that work? How does this bacterium make and detect this molecule? And so using genetics and molecular biology, we found the components of the circuit. And so now this is again supposed to be my Vibrio fischeri cell. And what we found was there was an enzyme, a protein inside of the cell that makes that hormone, the autoinducer. And that sneaks out, it just diffuses outside of the membrane, and then it accumulates outside of the cell. And then sitting on the cell surface, on the cell membrane, is a receptor protein. And this is just like the receptors that sit on the surface of your cells. And so they sort of have a part on the outside of the cell, and then that connects the outside world to the inside world. And so what happens is, as this hormone accumulates, this autoinducer, it can slot into the receptor, and when the receptor binds, when it connects to the autoinducer, that flips a switch and information from this receptor comes into the cell to tell the bacterium to turn on the genes that make the enzymes that make light. And so it's really simple how this works. The more bacteria there are, the more of that autoinducer there is. At a particular threshold amount, it will bind the receptor, and then all the bacteria will turn on light. And so that's how they communicate. Their word is chemical, right? And so you might think, okay, so why'd you get the plaque, right? So this glow-in-the-dark bacterium communicates. Well, it was originally found in this bacterium because we could just see the light these bacteria turned on, right? You can't see bacteria. They're invisible. So if they're doing things alone or doing things together, how would you know? You can't see them. You can't see their behaviors. But we could see that light. And so then with that hint, we and other scientists started to look at all kinds of other bacteria, and we found that the genes that were in this glow-in-the-dark bacteria were in hundreds and hundreds of species of bacteria. So now we know this is not an anomaly of this Vibrio fischeri, that in fact this ability to be able to communicate and act as a group is the norm in the bacterial world. So now we have hundreds and hundreds of species of bacteria that all have an enzyme that makes an autoinducer. There's always a partner receptor protein, and then together these components turn on all kinds of genes that bacteria want to express when they're in communities, but not when they're alone. And so now we have a fancy name for this field. We call this quorum sensing. So the bacteria are counting to a quorum. A quorum means the number, hey, it's a holiday, the number that you need to do something, and that's when they all act. And so the kinds of behaviors that bacteria control with quorum sensing are behaviors that you need lots of cells acting together to make the behavior effective. So the big deals in my field are that bacteria control biofilm formation, which is how they sit on surfaces in communities, which is how we know bacteria live in the real world now, and they are a scourge in medicine, right? Because you can't get rid of biofilms, so they cause persistent infections. And so the bacteria sit on surfaces, and then as groups, they release their virulence factors. Right? And so now what we get is if the bacteria can't talk or they can't hear, they can't make biofilms, and they're completely avirulent because they cannot undertake these group activities. And so if you think about it from the bacterium's point of view, especially having to do with virulence, the stupidest thing a bacterium can do is to, if there's only one or a few bacteria, is to release their virulence factors, right? They can't do enough. A single bacterium can't release enough virulence factors to make me sick. And furthermore, your immune system is trying to hunt them down. So the better idea is for the bacteria to wait, to count themselves with these small molecules, to recognize when they have the right number, and then if all of them launch their attacks together, they can overcome a bigger organism's immune system. So now we get that virulence and biofilm formation, which is the first step in virulence, are controlled by these quorum sensing circuits. Right? So these are broad spread in the bacterial world. So that's how the circuit works, but of course the bacteria are not actually talking with triangles, they're talking with molecules. And so we started to do some chemistry to try to understand, well, what are these molecular words that the bacteria use? And so the first molecule that was discovered was the one from this glow-in-the-dark bacterium, Vibrio fischeri. So in my cartoons, it's a triangle, but this is the actual molecule that is represented by that triangle. And so then we and lots of other scientists started to look at all kinds of different bacteria. And so these are just some different quorum sensing bacteria and the molecule that each one of these talk with. And what you can see is that the left-hand part of the molecule is identical in every species of bacteria, and then these right parts, these are just carbons, are a little bit different in every single species. And what those tiny differences do is to confer exquisite species specificities to these molecules. So what I mean by that is that Vibrio fischeri can't respond to the Vibrio harvii molecule, and likewise, if we put the Vibrio fischeri molecule on Vibrio harvii, it wouldn't respond. So these receptor molecule interactions are like locks and keys. They're perfect, and there's no slop in these systems. So what we understand then is that each of these molecules allow intra-species communications. This is how I count my siblings. Right? So they're private, secret conversations that a particular species of bacteria carry out to count how many of its family members are there. And so once we got that far in our studies, what we started to think about is that that's really great if you're Vibrio fischeri and you live in this pristine environment of this squid, but in fact, most bacteria don't live like that. Most bacteria live, as I told you, in biofilms, so attached to surfaces, but they live in mixed species. So this is a picture taken under a microscope of human skin. So this is a bacterial biofilm that would be on your elbow, and what you can see is that your skin is covered with bacteria, but the point from this slide is that every different shape or size is a different species of bacteria. And so what we started to wonder is if quorum sensing is really about counting your neighbors, and these molecules are so species specific, how do bacteria count when they're in mixed species communities? And so we wanted to explore that question in my lab. And so what we decided to do is to use a different bacterium named Vibrio harvii. It's related to Vibrio fischeri. It's a bioluminescent marine bacteria, so we could measure light production, but it lives a very different lifestyles. It lives in biofilms in mixed species outside of a host. And so we thought, so how does this bacterium do quorum sensing? And so what we did was we made mutants to make Vibrio harvii cells that were defective in quorum sensing, and just like I told you for Vibrio fischeri, we found the components by doing genetics and biochemistry and chemistry. And so what we found when we looked at this free-living bacterium's quorum sensing system is that we found an enzyme that made a molecule and a partner receptor. I actually already showed you that molecule and pointed it out to you. So we found a system for intra-species communication. But what we realized was that if we got rid, through mutations, of this system, this bacterium still had quorum sensing, and it could still count. And so we went hunting using genetics, and what we found was that there was a second quorum sensing system. So there was a second enzyme that made a second autoinducer molecule. It had its own partner receptor, and then together, the information from both of these molecules comes in to tell Vibrio harvii to turn on all kinds of genes at high cell number, some of which are bioluminescence. So bioluminescence and many other group behaviors. And so what we wondered when we got this far is, what's the point of making two molecules? Right? If these molecules don't encode different pieces of information, having two shouldn't be better than having one molecule. So we wondered, what's the point of this second system? And so what we did was we purified this molecule and we found the genes for it, and we looked in all kinds of other species of bacteria. And what we found was, although this system and this molecule was very species specific, as I told you, only Vibrio harvii seems to make this molecule, every bacterium that we tested had the gene for this enzyme and made this molecule. And so we purified that molecule, and this is it. We call it Autoinducer 2, because it's the second one that we discovered. And you don't, all you have to know, it's 5 carbons, it's got all these oxygens on it, and the only point I want to make about it is no matter what bacterium we purified that molecule from, they all made the identical molecule. So this is different than the first system, where each bacterium makes a slightly different molecule. In the second system, they're all making the identical molecule or the identical word. So in fact, what that means is that this is a non-species specific language. This is the language of interspecies communication. So I can talk to you and you can talk to me because we have the same molecular language. So this is sort of the universal bacterial trade language, if you will, that all bacteria seem to use. And so now what we think is that bacteria have to at least be bilingual. So they have to have some molecule that says, this is me, and then they use this autoinducer 2 to say, this is other. And so the computation that we know bacteria do now, what we think they do is, the first thing they do as they're out in the environment is they just scan the environment with these receptors, and they ask, are there any quorum sensing molecules there? And then based on whether or not there are molecules, they begin to turn on group behavior genes. But then the more sophisticated thing the bacteria do is that they actually measure the ratio of those two molecules. And I think what they're trying to do, they're saying, are there more of me and my family, or is there more of the enemy around? And then they change which behaviors they turn on based on whether they and their kin are in the majority or somebody else is. Right? So in fact, they can tell self from other, and they behave accordingly based on who's winning and who's losing. And so, of course, I've already alluded to the fact that these quorum sensing systems are widespread and they control virulence. And so now that we've discovered that bacteria have communication and we've discovered that it's vital for making infections, the sort of applied part of my field now, which I wanted to finish with, is wondering, can we interfere with those communication circuits to shut down virulence? And so probably you know we have this global problem with antibiotic-resistant bacteria. There haven't been new antibiotics made. The bacteria are becoming increasingly resistant. It is an urgent problem of global importance. Right, what we're wondering is, could you make a brand new kind of antibiotic? So traditional antibiotics kill bacteria, right? What we're wondering is, would it be enough to make them deaf or make them mute, right? Just so they don't know to turn on their virulence cascades and let the immune system get rid of them. So it would be a completely new paradigm for thinking about antibiotics. And so we want to try that. And so there's sort of two strategies. One is if you wanted a narrow spectrum anti-quorum sensing therapeutic, you would try to interfere with the species-specific system. Like if you wanted a medicine for one kind of bacterium, like Pseudomonas, right? Or let's say you're sick and your physician doesn't know what's wrong with you. Then maybe what you'd like to do is to have a broad spectrum anti-quorum sensing molecule that works against many species of bacteria. So we're trying to make both of those, and what I wanted to do is to just, I can't help myself, tell you our newest results. So I'm going to tell you one story about trying to interfere with this species-specific molecule. So you can't do this in Vibrio harvii. It's beautiful, but it's never hurt anything. So you can't say, can I make a molecule that turns off virulence if you're working in a bacterium that's not a pathogen, right? So what we had to do is to switch to a terrible pathogen, which is called Pseudomonas aeruginosa. This is the bacterium that is sort of rampant in hospitals. Like you go in to get your gallbladder taken out, and you come out, you get a Pseudomonas infection, right? Because they're very sticky. They love to make biofilms on equipment in hospitals, on catheters, on intubation tubes. They also are opportunistic pathogens, so they kill people that are immune compromised or have burns, and they also kill people who have the disease cystic fibrosis. So probably you know this disease CF, or cystic fibrosis. People have a genetic mutation in their lungs, so they can't clear their lungs. What happens is they become permanently colonized with Pseudomonas aeruginosa, and that's what kills people who have CF. So it is a terrible pathogen, it's multi-drug resistant, and it's a real problem in disease and just in medicine in general in hospitals. So we wanted to see whether we could turn off quorum sensing in this bacterium to stop virulence. And so again, just to remind you, obviously Pseudomonas has quorum sensing, and again, it turns on biofilms. So Pseudomonas sits on surfaces and makes communities, and then it launches and releases all of its virulence factors only at high cell number. So this is the molecule that I already showed this to you. This is the molecule that Pseudomonas talks with. And so what we did was we made a molecule that looks kind of like the autoinducer. This is an inhibitor. So if you look at it, the left part of the molecules look kind of the same, but there's a big bump on the end of our molecule. So this molecule, when it slots into that receptor, it jams the receptor and it prevents the receptor from detecting the real molecule. So this is called an antagonist or an inhibitor, right? And so it's like a key that's stuck in the door, but it can't unlock it. And so we made that molecule, and now we want to see, can we shut down pathogenicity in Pseudomonas? So remember I told you the first step in virulence is the bacteria make a biofilm, right? So they spread out across the surface. So what you're looking at here, this is a Petri plate, and what we did was we just put a little bit of Pseudomonas right there in the middle on the top of the Petri plate, and then if we wait overnight, it crawls out to the edges and it makes this beautiful biofilm. So if we put that inhibitor in the Petri plate, it stops it. So that was good news. So step one is we can stop Pseudomonas from making the biofilm. Okay, so that's step one. Step two is the launching of these, release of all these virulence factors. So we wondered if we could shut that down. So what you're looking at is that experiment. So wild type Pseudomonas makes a terrible virulence factor that's called piocyanin. That's what kills people's lung cells. And it's called piocyanin because it's cyan. It's blue. It's a molecule that has a color. So when Pseudomonas is highly virulent and it's at high cell number, it releases this piocyanin virulence factor, and the whole culture turns green or blue. What you can see is if we make a quorum-sensing mutant, it's colorless. That's because these bacteria can't talk, so they don't release the virulence factor. But if we take the wild-type cells and we add the inhibitor, what you can see is we can shut down production of that virulence factor by 90%. So now we're in business. We can stop biofilms and we can stop the bacteria from producing these virulence factors just by making them incapable of communicating. So the next question is, you can do it in a test tube, you can do it on a Petri plate, can you do it in an animal? So there's an animal model for Pseudomonas, and so we got that up and going in the lab. And so what you do is you just ask, it's easy, are the animals alive or dead, right? And so if you don't give the animals Pseudomonas, the animal is perfectly fine, right? It will live for a long time, right? If we infect the animal with Pseudomonas, what you can see is all of the animals die in a day. So if we do that experiment, but we give that molecule, that synthetic inhibitor that I just showed you to the animal, in fact, it's as if you didn't infect them. So now we're getting somewhere, right? And this idea that there's merit behind this idea of making anti-quorum sensing molecules to stop infections. And so this is where we are. This is not a medicine, right? You'd have to take a pill this big and it would kill you. We have to actually make this molecule much more potent. We have to make it look more like a medicine. We have to get it where we want it to go. We have to give it a good lifetime. And so now what we have to do is work with medicinal chemists to take that molecule I showed you and actually turn it into a medicine. And so that's what the lab is doing right now. And so that's a little summary of my lab's work. I'll just remind you what I already told you. I hope what you think is that bacteria talk to each other. They don't have voices. They use molecules as their words. They are multicellular. So remember, you already heard from Mr. Lynford, these bacteria have been here billions of years. Humans have been here a couple 100,000. You know, bacteria invented or evolved multicellularity. And so we think that by learning how these bacteria undertake these group behaviors, we'll be able to understand something about the evolution of higher organisms and how other groups of cells or organisms work together, because the principles will be embedded in these ancient, tiny creatures. Of course, with the molecules I showed you, you can tell that bacteria can distinguish self from other. Again, that's a trait that happens in higher organisms. It's not like your heart and your liver get confused every day. That's because different molecules carry out different jobs and tell these organs what to do. We think, again, by studying bacteria, we're going to learn about how those processes work in all organisms. And then, of course, the applied part of the story is to develop strategies to stop quorum sensing and harmful bacteria, but to go back to the beginning of my talk and what you're going to hear from Dr. Blazer, right, is that most bacteria that we encounter are working as our 24/7 partners to keep us alive. And so maybe the real way forward in medicine is to make pro-quorum sensing molecules that make these bacteria chit-chat even better, and those could be, we could actually enhance the beneficial bacteria instead of fighting against the harmful ones. And then the final thing to tell you is to finish with a confession, which is that we didn't think any of this up, right? When now, you know, that we're looking for these pro and anti-quorum sensing molecules, we started thinking, you know, well, these bacteria have been fighting out in nature for billions of years. Maybe they already made these molecules. And so sure enough, if you go out and look in natural environments, there's bacteria that eat each other's autoinducers. There's one bacteria that makes an enzyme. The guy next to it cuts the ring open. So this one guy is trying to talk. The guy next to it in the dirt makes it mute. They make antagonists. They eavesdrop. They free ride. They do all these shenanigans. And so, of course, those things that are out there have risen over billions of years of evolution. The idea that I and a 22-year-old chemist are going to make a better molecule is ridiculous. And so what we should just do is go find those strategies and bring those into the lab and tinker with those, right, to make them even better. And so there's a lot of people working in that space right now. And then let me just finish by showing you the gang that's 40 miles down the road. This is my lab, the lab people in Princeton, and what I want to say to the students is that they look just like you. Whenever you hear about something new or amazing in science and engineering, right, the chances that someone your age made that discovery is really high. So at least in this country, science and discovery is done by young people. And what I would also say, besides the fact that you can do it, what I would also say is that being a scientist is the greatest life in the world. I mean, we get to go in, we make discoveries, we get to think about things that no one has ever thought about, right? We hope that by understanding these fundamental processes, we'll actually do something good for mankind. I think my lab is getting there, right? But it's just fun and it's exciting and you're with smart, creative, energetic people all of your life and you're trying to actually change the world, and the biggest problems that are facing us are biological. Their health, their food, their energy, their environment, all of those will have biological solutions coupled to engineering and coupled to chemistry and coupled to applications, right? And so if you're one of the students, I hope you'll come talk to me afterwards because I'd love to tell you about how it is to be at least a biologist and just to remind you that I didn't make these discoveries. I type. I give talks, right? But people your age did, and it's really, really fun to be in this business. Anyway, and so that's my talk, and maybe we want to do talk here. Want me to just go somewhere? This one? This one? Yeah, thank you.
How enthusiastic is that? If you can't be excited after hearing that talk, you must be asleep. So I think the concepts, now we have to figure out how to take it from her chemistry lab and take it into the real world, which is part of the reason we have these two interlocutors, because they are looking at it in larger bodies like ants and bees. They're looking at it with what is the downside of microbes in the body, and I think that's where we'll start, but I'd like to introduce the two of you a little, if I may. It's okay, doctor and doctor? Dr. Cronauer, Dr. Bassler. My right, your left. We'll start with Dr. Bassler. And I get a commission for each one of these books you buy that he has just come out with. And it's really a fascinating book. It's called Missing Microbes, How the Overuse of Antibiotics is Fueling Our Modern Plagues. Clearly, Big Pharma is not one of your enthusiastic supporters. Question?
They don't care.
Yeah.
They don't care. Okay. We're going to talk about what that means, but it's interesting. This bastion of liberal journalism, the Wall Street Journal, last Tuesday, that was the 11th, and then on the 7th, have articles that are very relevant to what he does. Purdue chickens, now free of antibiotics. They've eliminated all antibiotics from their chicken supply. And they're the first major poultry supplier to do so. It's taken them 14 years to figure out and to do it. And they impact now 13 million chickens. So next time you go out to McDonald's and have a chicken McNugget or whatever it is, think about how healthy it could get. And then this new study, which was yesterday, finds that allergy risk starts in an infant's gut, the bacteria that's there. And I'm not going to steal everybody's lines, but I want to tell you something about our eminent Dr. Bassler, if I may. And your wife is here, so I can embarrass you, right? All this good work that he's done, I'm going to try to say in 5 minutes. He is the Muriel and George Singer Professor of Medicine, Professor of Microbiology, and Director of Human Microbiome Program at NYU School of Medicine. And before that, he was the chair of the Department of Medicine at NYU from 2000 to 2012. So he's one of us, and he's now gone from the dark side, which is administration, and back to the lab so he can do something value added for society. He graduated in 1969 from the University of Pennsylvania. 73, he got his MD degree from New York University. Between 73 and 79, he did residency and fellowship at the University of Colorado in internal medicine and infectious diseases. He is the president of the Infectious Diseases Society of America, the chair of the Board of Scientific Counselors of the National Cancer Institute. He's the chair of the Advisory Board for Clinical Research of the National Institutes of Health. He was elected to the National Academy of Medicine and the American Academy for Arts and Sciences. He holds 28 patents and has authored over 550 original articles. And in 2015, Dr. Bassler was designated as one of the 100 most influential people in the world by Time magazine, and he was appointed as the chair of the President's Advisory Council on Combating Antibiotic-Resistant Bacteria. So very, very impressive. And you're also here, homegrown, so we're really excited about that. And maybe I should take this time to talk a little bit about Dr. Cronauer here, who sits to Dr. Blazer's left, and he's very young compared to Dr. Blazer, right? You're the youngest.
Yeah, sure.
Yeah. So he's the head of the Laboratory for Social Evolution and Behavior at the Rockefeller University. And so I'm having Chinese food earlier last week, and it turns out that a good family member of ours works in the lab across the way from you, Meg Younger. And when she heard you were speaking here, she was so excited. So we decided whoever sees you first says hello. So I'm saying hello from Meg. And so your reputation precedes you. And he received his biology diploma from the University of Würzburg in 2003, his PhD from the University of Copenhagen in 2007. In 2008, he did postdoc work at the Universities of Copenhagen and Lausanne. Between 2008 and 2011, he was a junior fellow at a school called Harvard. And that's very prestigious because they only allow 12 recipients per year. He speaks, now get this, 6 languages. German, which is native, Danish, English, French, Latin, and Spanish. Now that's really exciting. Now, he also specializes in working with social insects like bees and ants, who may use quorum sensing. So I'm going to take a breath for a second and say, since you know this young lady so well, why don't you talk about or ask her where your two worlds intersect?
I've heard Bonnie speak many times, and I've learned something every time. It's just amazing. And I will give a little anecdote, and that was that in 1998, we started doing some work on anthrax, which was very timely. And in 2001, we know about the anthrax problems, and we decided that we'd start to do some work. We wanted to work on quorum sensing and see if the bacteria that caused anthrax had a quorum sensing system. So I called up Bonnie and said, Bonnie, can we have some of your Vibrio harveyi strains that you made, your reporter strains, so that we could use them as controls? And she immediately sent them, and we almost immediately found that B. anthracis had that same AI2 system, which wasn't surprising, but we began a line of work on that. And in fact, we did some work on some natural products that algae make, furanones, a certain kind of molecule, to see if we could inhibit anthrax using these molecules. And we could inhibit them very well in the test tube. And then we worked in mice, and unlike your beautiful results, we had one of the best mouse models that I have ever developed, in which we could reliably kill the mice with a vaccine strain of anthrax, and we could rescue the mice with antibiotics, and the furanones had absolutely no effect. So we were disappointed, but we went on to other things. But I wanted to mention one of Bonnie's great traits is that she's a very generous scientist. She shares her work, she shares her reagents, and that's a very useful and important characteristic.
So do you worry about some of the things that she might invent or find actually being antithetical to what you're trying to do?
So then the next time I called Bonnie, it was a few years later when we were doing a lot of work on the microbiome. So the microbiome is all the bacteria, all the microbes that live in our, let's say, the human microbiome is all the microbes that live in the human body. And we were very interested in antibiotics and how antibiotics perturb the microbiome. And it seemed to me obvious, although not proven, that there was going to be a lot of quorum sensing in the body, bacteria talking to other bacteria, especially in the gut, where there are huge populations of bacteria. So I wanted to try to study this. And so I went back and called Bonnie and said, can you help us study this? And a student from my lab went over to Princeton and relearned some of the methods. But in our case, the problem was too complex, still waiting to be solved, because it's easier to work with pure cultures. than mixed cultures where you have hundreds or thousands of species. And that's kind of where the problem is. So I believe, and maybe Daniel or Bonnie could comment about what we know about quorum sensing in vivo between species of bacteria in a host. I believe that just as you say, that's going to be a useful target to either enhance what we have. It's hard to enhance, but maybe to inhibit some of the bad guys. Because with antibiotics, we don't have to kill the bacteria. We don't have to kill all the bacteria. We just have to, and for the engineers in the audience, it's all a problem of rates. It's about the rate of multiplication of the bacteria versus the multiplication of the immune system. So if we can slow down the bacterial multiplication, then the immune system gets ahead. We don't have to kill them, we just have to inhibit them effectively. I'll stop there.
Well, maybe what we'll do is turn to Dr. Kranauer and say, given your work with social colonies, does quorum sensing have a role in any of your work?
Yeah, so there's certainly a few cases where quorum sensing happens in social insects as well. Of course, I always get jealous when I hear people who work on kind of social behavior, social evolution questions that work on bacteria, because it's so much easier to study them. They have like very short generation times. You can actually do genetics. So we don't know that much about the molecular details yet when it comes to social insects. But I would say one of the classical examples are honeybees. So honeybees, when the colonies reproduce, they split in two, and then one of the two halves leaves the nest, and they have to find a new nest. And how they do this is they swarm, they form a swarm, and sometimes they put the swarm at your balcony or something. Like often, I think the fire brigade is being called because of honeybee swarms that hang on somebody's balcony. And then what they do is they send out scouts that look for new possible nesting sites. The scouts come back to the swarm and they perform what is called a waggle dance, where they basically communicate the distance and the direction of the nesting site they've been assessing. And they modulate the waggle dance depending on how excited they are about the new nesting site. And the other bees in the colony, they assess that and they try to recruit other bees to the same nesting site to assess that nesting site as well. There are other signals involved as well. So for example, there's been one signal that has been discovered quite recently where if a different bee dances and advertises for a different nesting site, if a bee is really excited about her own site, she can interfere with that dance. So there's all these kind of interesting feedback loops. And then at some point when the bee swarm reaches a quorum, the kind of signal changes once again, and then the bee swarm takes off and moves to the new nesting site. And it turns out that bees are very good at collectively choosing the best available nesting site. So this way of reaching a quorum actually works tremendously well. There's a very interesting book by Tom Seeley called Honeybee Democracy about this phenomenon. It's really a very nice read. And he talks in this book like how he applies what he's learned from honeybees to faculty committee meetings when they perform job searches. So it's a very direct way of reaching a consensus in some ways. Actually, what I had to think about, and I don't know whether that has even been discussed as a quorum sensing phenomenon, but I think it is, was when Dr. Bassler talked about the two different types of quorum sensing, like once within the own species, and then also the mechanism of assessing the density of bacteria of other species. There's something somewhat similar, I guess, in the system that I worked on for my diploma thesis, which are honeypot ants that occur like in the southern US and northern Mexico in the deserts. And they're called honeypot ants because they have these, like a worker caste that is basically filled with food, and they're so blown up, so fat that they can't walk anymore. The ants just pin them to the ceiling of the nest. And then when the colony gets into dire straits, they take down the honeypots, and the honeypots regurgitate food and they feed their nestmates. But they also they're very territorial, so they form these tournament sites between colonies, and it's a very ritualized tournament. So usually these tournaments don't escalate. There's no real fighting going on. But what they do is they recruit the largest workers to these tournament sites, and then they walk around and they can tell which ants, whether an ant they encounter is from the same species or from the same colony or from a different colony. They do this via pheromones, where chemicals sit on the cuticle of the ant. So it's somewhat similar to how bacteria do it, apparently. And they have some way of measuring a ratio of ants from their own colony and ants from the other colony that they encounter. And the size also plays a role. So they actually walk around on these like stilt-like legs, and sometimes you see them do their displays on little pebbles or rocks. So they kind of fake it a little bit, like they pretend to be even bigger than they are. And so they have some way of integrating, like how many ants from their own colony are there, how many ants from the other colony are there. And then if one colony, if the colonies are similar size, often the tournaments subside and there's absolutely no escalation of fighting in any way. But if one colony is larger than the other, then the tournament side slowly shifts towards the nest entrance of the smaller colony. And at some point, the colonies can reach some quorum at which the behavior changes from these ritualized tournaments, and the larger colony just raids the smaller colony. And they actually capture the brood of the smaller colony, transport it back to their own nest, and then the ants from the other colony then hatch, become mature, and they work as slaves. They have no idea that they are in the wrong colony, basically.
Egypt, or maybe.
It's awesome.
So this is to say that I think these quorum sensing mechanisms are every time you have like some form of social behavior, and that's of course common across all different scales of organization of life. These kind of quorum sensing mechanisms can become important in one context or another.
Now, I don't have microphones for the audience, but if someone has a question, they raise their hand and speak very loudly for our guests. Dr. Rubenstein.
So I wanted to go back out of curiosity to the
Speak loudly so everybody can hear you.
I want to go back to the quorum sensing, which created when they all reached
Just leave it there.
the light. And the question, I guess, that I have.
Stand up so they can see you.
Okay. Is there some evolutionary principle in your mind about why they wanted to create light? And then that's, I guess, the first piece. And then second question is, how would they have decided, either species, that it would be useful to have the light? In other words, how would they capture that idea?
Right. So, so in the ocean, light's a big deal. Right, so on land, there's only a couple organisms, fireflies, there's a fungus, there's a glowworm that make light because we have light, right? So, but in the ocean, it's estimated that 75% of all the organisms, bacteria and higher organisms, either make their own light or use somebody else's, because you don't have to go very deep and there's no light. So light's a big deal down there to see, to signal, right? So it's used all over the ocean. So that's very, very common. So you just have, it's like Christmas down there. All the light is blue, like what you saw on my slides, because that's the color of light that travels the farthest in water. And so that's that. And so then, but your question is, why is a bacterium making light, right? And so the idea is that originally that probably, so the bacteria are the oldest organisms in the ocean too, that probably the light is a byproduct of this reaction, which actually does other biochemistry, and then and it releases heat as light, right? And that was just a byproduct. But then, if you get a free ride for it, right, and then you get to eventually live in this squid, and you get to do this, get all these goodies for it, over evolution, you'll keep it. You only have to have a tiny, this guy should be talking about it, a tiny advantage, and over evolutionary time, that property gets kept, right? And so I told you the one about the squid. There's all kinds of these different associations, like angler fishes. That's Dubrio fisheri, and they hang that lure out, and some animal sees that. And then there's flashlight fishes that you've seen. So all of those are these different symbioses where the bacteria are getting fed for making the light. And so the ocean, free living in the ocean, the way Vibrio harveyi does, the second one, that's a nutrient desert. The good life is in this light organ of this squid, or these, because you can make many more of yourself. And so the real question for us now is, why do the free-living ones still have it, right? And that we can't really answer, except that it's this byproduct of this reaction, and it's still just there. And maybe occasionally they get eaten and they get taken somewhere else, or they get to live in the gut, because they can live in the gut, and so they actually still get a benefit from it.
Yeah, I want to ask Bonnie a question and half answer your question, which is maybe the difference between engineering and biology is biology only has one principle, and that's natural selection. Natural selection is everything. All the rest is commentary. And so the conventional the conventional answer to your question is one day a chance event happened, and some light, presumably the bacteria that make light preceded the squid, because they're older, and somehow there was an association, and that squid lived longer. Maybe all of its compatriot squids who didn't have the association with the bacteria, they were all killed by predation, and this guy lived, and now they could pass on their progeny, etc. But there's a very sophisticated system with the development of the light organ. It wasn't just on the skin, but a whole organ developed, and the organ has many very specific properties to care for and feed this. Once the organ develops, you can imagine how it evolved. But what's the thinking about this?
So, right, it's like a camera. I mean, it's an amazing organ. And then also I'm not going to answer Marty's question because I don't know it, so I'm going to look over there. And but also it's the only bacterium in there. When Vibrio fischeri gets in there, all other bacteria are excluded. You never find a dark mutant in there. Somehow the squid can get rid of cheaters, right? And so so it's actually not known. And if you actually grow the squid in the lab without bacteria, the organ never develops. It's like it just stays this little miniature organ.
What happens when you put a dark one in? It's removed?
It's removed. Yeah, and so I don't know the answer to his question. He's right. It didn't happen overnight that the bacterium encountered the squid and then all of a sudden there's this light organ. But over time it did, and I don't know the answer to that.
You bring up a point that touches all of us, and that is that in any system of cooperativity, the downfall is cheaters. And yet nature has figured out how to deal with cheaters because we have all these cooperative systems in insects and pollinators and the like.
So that's for sure. Even not the squid, just quorum sensing, right? It's easier for me to think about for the reasons you said, right? These bacteria, they make these molecules, they do all these group behaviors. Like, just be a guy that doesn't do the group behaviors, right? So somehow there's little molecular police in this, right? That keep cheaters down, because otherwise these guys would get exploited, right? And so there have to be, as Marty's saying, mechanisms so that there's probably cheating, but it stays.
Who's getting rid of the cheaters, the other bacteria or the host?
Host. Yeah.
How costly is this?
So this is very costly. So to make light is really costly. So it takes 20% of their oxygen and all their energy to make those photons of light. Right? It's a big deal.
So if you compete strains in the lab where one doesn't
strains in the lab, a dark strain with a bright strain, the dark strain will win.
Yeah.
In the lab.
Instantly in the lab, right? Because, yeah.
We have two questions. You want to give Srini the microphone, and then we'll turn it over to my bride.
One of your slides showed a heterogeneous mixture of bacteria. Presumably, they don't have the same bioluminescence when you have this mixture. How do they communicate in such an environment? That's my first question. Sort of related to that in some way is, are there other kinds of collective behavior that is not bioluminescence, maybe that come into prevalence when you have the heterogeneous combination of bacteria?
Yeah, absolutely. So each species of bacteria has its biology, that it needs to do. So on your skin, which is the picture I showed, there are no bioluminescent bacteria. And so those bacteria, every species turns on its own set of genes that it needs. Like, so pathogens turn on virulence factors, biofilm formers turn on, biofilm production, they turn on different kinds of metabolism. And so all kinds of behaviors that you need lots of cells working together tend to be controlled by quorum sensing, way beyond bioluminescent, right? And so, the ones we've tended to study are things like virulence, right? So the bacteria count, and then they all secrete their toxins in unison, because that's the effective way to make an infection, right? And so each bacterium understands its quorum, like what number do I need to count to, and then it understands what genes it needs to turn on for its niche.
So before you take the microphone, where do you see your work going? When is there going to be a commercial application that these people can figure out how to deliver? Tell us, what's your vision thing of where your work is going to lead so we as human beings can take advantage of it?
So I think as about the medicine, the thing I showed a bit, that's actually sort of the hardest, right? You have to get it where you want it to go. It has to work. It's not allowed to make the person sick. So I think for the engineers in the audience, other applications are things like hanging these molecules off of materials. So that's happening right now. Like if you could hang anti-quorum sensing molecules off of plastics, you could make resistant-free, infection-free catheters or surfaces. People are trying to make antiseptics that have anti-quorum sensing molecules, for health, for hospitals. And so I think there's all kinds, people want to put them in agriculture. They want to actually deploy them in things like fertilizers, and then also making what I would call biologically inspired materials. That's a big deal, right? And there's all kinds of applications that one can think of to either get good bacteria to come or to keep bad bacteria off of them. And those applications will happen faster than medicines because you don't have to.
And are you subject to the same type of the criticisms that, let's say, Monsanto gets for creating engineered microorganisms?
So always, I hear like you're interfering with nature, and I think, yes, I am. That's my job, right? Is to actually figure out nature and try to learn something and then make something useful, right? And so what I would say to your question is, so the part of the adventure that my lab is in is the very first step. So our sweet spot is to make the discoveries. And to make the ideas. My lab will not make that medicine. We will not make biologically inspired materials. What we hope is we give the fodder. We give you the molecules, we give you some of the principles, there's ideas, and then people like you guys that are actually expert at a different part of the pathway, right, will make them into practical solutions, because this whole continuum of the pathway has to happen to get products, right? And I'm not good at the second step, I'm good at the first step. Right? But I think it's not far away. I mean, people are putting these in contact lenses now. There's ideas about, using them, as Marty said, perhaps as probiotics, putting them in lotions, stopping dandruff. These kinds of applications that I think are before the medicine will give us so much information about how to really make that medicine too. Right?
Can I maybe ask a follow-up
Go
question?
for it.
So for the applications when it comes to antibiotics, the big problem is, of course, evolution of resistance, right? Is there a reason to think?
Yes. Okay. So yeah, and Marty, so these guys are so right. So the big deal, right, is when you give a traditional antibiotic typically kills bacteria, and in any population of bacteria, there's mutations. And so if somebody has a fortuitous mutation, you kill off everybody. The fortuitous mutation that makes it resistant, it and its offspring live, right? Because you killed off all the competitors, right? And so that's how we get these problems with antibiotic resistance. They're pre-existing mutations in the community. You kill the guys that don't have those, and then you basically give real estate, you allow the others to grow. And so the idea with an anti-quorum sensing molecule, right, and we've actually made mutants that are resistant, right? Like these guys are trying to talk, they're trying to do this collective behavior. If one individual in the population is resistant to the anti-quorum sensing medicine, right, it turns on its quorum sensing cascade, it makes all its toxins, it's going, come on, you guys, come on, but nobody else is participating. And so you don't get the collective behavior. So it turns out it actually has a growth disadvantage, because it's like, come on, you guys, come on, we should be doing all this, and everybody else is impervious. And so that's, at least in the lab, the idea is that these could have, the bacteria will get resistant, right? They do evolution on a very fast time scale. But the idea with an anti-quorum sensing medicine is you would get a long shelf life. You would just get longer for somebody your age to figure out the next trait to go after.
Tondra?
Oh, my question, does it have any, is there any difference when the host of the bacteria is older or younger? Is there an age factor in how all of this happens?
So my stuff we don't know about, but maybe you want to talk about that.
I want to go back to the last question. The question about whether resistance will develop or not is a really important question. And I think many people who work in new fields say, oh no, resistance will never develop. Fortunately, Bonnie's a very honest person. She understands that resistance likely will develop, but the real question is, what's the cost of resistance? Because resistance has a cost, and if it's too costly, it will develop, but those guys will be so hobbled, they'll be in such bad shape that they will never come about. So I think it's still an open question. If you really developed good anti-quorum sensing molecules, what will the resistance situation be? And it will not be the same for all organisms. Just as there are bacteria today, 70 years after antibiotics, that still are not resistant, and there are some that became resistant within months of first use. And in terms of age of the host, which was your question, what we know is that when babies are born, when they're in the womb, they're essentially free of bacteria, as far as we can tell, of living bacteria. And then the big delivery of bacteria is when they go through the birth canal. When they're born, they are acquiring a lot of bacteria from their mothers. Their microbiome is very different from the adult microbiome. It's very different. And then it gradually morphs toward adults. The first 3 years of life are the really critical time. And then we don't know a lot. After that, we know that it's quite different and eventually becomes quite similar. And there are different populations. There are ecological successions, just like we learned about successions in forests. And I'll bet quorum sensing is involved in many of these processes. We just don't know.
So, right, because I think that's sort of one of the and Marty already touched this, the fantasy is to actually up the complexity, right, and actually begin to, maybe we know enough about quorum sensing now to be able to take on these really complicated, thousands of species, they're in different space, what you ate, your human genetics, but I think, so for my field, that is the horizon for the next 10 years, is to, we shook them around in a flask and we found some parts, but now the idea is to really take those studies into scenarios that actually mimic something much closer to the real world. And so I think that lots of people in my field are moving in that direction. So hopefully we'll know more soon.
So let's end with, so there's some time for, I'm going to take your question in 1 second. So what's going to happen is we're going to end, and there's going to be a reception out there, and you can talk with our guests one-on-one. And why don't we have your question, and then I'll do a wrap-up.
How fast do they communicate? Is there a unique communication pattern that enables fast communication?
And before they answer that, we're going to have to say it again so we can hear you. But tell a little bit about yourself. What year of student? What are you studying?
Okay, I'm a senior. I study computer science, and I deal with networks and consensus in networks. So I guess this is a type of consensus in nature, and I think designing systems from nature is important. So that's why I was wondering if there's a fast communication pattern that might be useful for computer networks to communicate quickly.
So it's extremely rapid. It's diffusion, right? And so the molecules, get between cells in milliseconds, right? And then the biochemistry to send the information from the outside to in is essentially instantaneous. Turning on genes and making proteins is slower. So the output is slow, like it's on the order of minutes, but the communication between cells is as rapid as diffusion.
Thank you.
There's another question out there from any students. There's right here. I'm going to bring the microphone down, and we're going to ask you to tell a little about yourself and before you ask your question.
Hello everyone, my name is Zhang Yu. I am now a freshman majoring in mechanical engineering. Technically, I know nothing about biology and biomolecular engineering, but I have read an article before at an international science and engineering fair about a round pinewood doorknob that is antibacterial doorknob, because people always get infected by the bacteria they got on the doorknob, and yet a student at Luxembourg found that when the doorknob is round and it is made of pinewood, it has the most effective effect of killing the bacteria, and so people don't get sick. And that is a work from the engineering student. And the first question I'm asking you is that how you see the two disciplines. One is natural science, is biology, and the other is engineering. How they could come together to accomplish some work. You have done some work about the quorum sensing, right? I know. Although the PowerPoint you've presented to us is just some short slides with simple principles, but I bet your PhDs, your masters have done enormous work on that. And every scientist, everyone who has graduated from college has known that every scientist have only answer a specific question about like how the bacteria communicate with each other. But we are having an undergraduate study studying multiple disciplines. I want to know how a freshman becomes a PhD to studying such a specific discipline, and how, there is a trend called interdisciplinary study. How engineering and biology comes together to form a solution.
Perfect question.
Yeah,
Go for
absolutely.
it.
So it is, I'll just say this, it's the future. You can't be trained the way I was trained and be successful. So where the big breakthroughs come are when disciplines meet up and people go beyond their comfort zone. So you're right, I told you, you're very perceptive, I told you this mini story, right? So I just told Tondra, we want to try to understand how does this work in your gut. So that means flow, it means space, it means microscopy. I have all kinds of physicists and engineers and material scientists, this is a material, right, working in the lab right now. They're learning the genetics, we're learning about flow, we're learning about eddies, we're learning about, all kinds of engineering principles, material scientists, and then everybody does better, right, by actually crossing over into disciplines. And so now, scientists and engineers get trained to think analytically, to be logical, right? The techniques are transferable. And so you just saw, the chemists found those molecules. Chemists made the medicine. The biologists found the genes. Physicists, where's the network guy? We have a physicist who models that circuit and how do you get rid of noise, how do you avoid cheating, how do you get fast, slow steps, and then the engineers are teaching us about flow and spatial constraints, right? And so they're all working in the same lab, they're fearless, and just like what you said, is that they believe you gotta come out of the traditional disciplines if you wanna be successful in the 21st century. So you should take a broad number of classes, they're gonna make you major in something, but that doesn't mean you can't learn something else and go across these disciplines, because it's the future.
Now here's what you want to do. You want to write down your name. Seriously. This is what I do for the last 40, 50 years to try to create my human network and give it to her. But don't wait 4 years for her to come back to you. Every 6 months, check in. Tell her something that you've read that might be of interest to her. Something that you might be interested in, ask her a question. A book that you might has just come out, or an article. Now you might not get a response as quickly as you would like, but on the other hand, if students take the time to seek you out and tell you something you didn't know, that says something to you. And so it's the human network as well as the scientific network. So as you leave today, you hopefully you have a couple of more tools in your kit. Any other questions? Okay.
Well, I just want to say that Bonnie just described quorum sensing in her lab to get to enlightenment.
Oh, brother. This young lady, maybe that little bit. Yeah, he has
Another gentleman here is raising his hand. When you get the guys in the middle to actually ask questions, we know we're making some progress.
Hello, my name is Flynn, and I am a biomolecular science major here. And
What year are you?
I'm a freshman too. And I have some questions about the half-life of autoinducers and how does it echo, how does it degrade, and does it make a good biomarker for diseases, etc.? Thank you.
Right. Yeah. So people would love to. So I think that's a really good idea. The second one, like could you use it as a biomarker? So lots of people would like to make a little dipstick, like a pregnancy test. But if these molecules are so specific, or the generic ones are really have to do with how many bacteria are there. There's a fantasy that you could use these. I should have said that when you asked about applications, but I didn't. You did. There's a fantasy that you could use them as diagnostics, right? Like you dip it in and the thing turns a color and you know you've got this bacteria or that. So I think that is a totally interesting way to go. And so they're actually, they're pretty, if, you know, in water or buffer, they're pretty stable, right? So it's not, they have, you know, they have half-lives that last hours to overnight, so you could absolutely do something like you're thinking. That's a great idea. Yeah.
Now, besides this energetic person to my right, to her right is someone from the Rockefeller University, which is one of the most impressive organizations on the planet. So now what you have to think about as you're sitting here is, what kind of questions you want to ask him to get his attention so he'll remember the question. Is there anybody that wants to put you on the spot that you have a question for him? All right, well, he's going to be back up there at the reception, so you're going to have a second chance.
There's a question in the back there.
Oh, is there? Come on down.
Good.
Introduce
Good
yourself.
evening, everyone. Excuse me, I'm feeling a little under the weather. My name is Samantha. I'm not a student, but I have 3 students here. So I have a chemical engineer, I have biochemistry, and a veterinary science up and coming. My question is, I'm actually a Christian and a scientist at heart. I really love science. I love coming to talks like this and finding out what the new discoveries are. I hear you talking a lot specifically about biomimicry. And obviously, biomimicry is something of recent, is something within the last couple of decades that scientists have been looking into as a viable option to study in science. It's very interesting that it's of recent. But I just wanted to say, as a Christian, have you guys ever considered, I understand that evolution is like the standard perspective, the standard view lens. Have you ever considered, with the understanding of how biomimicry has helped science within the last couple of decades, maybe transferring the lenses to a creationist kind of perspective, just to look at it from that side, to see how possibly the whole scientific discovery could be accelerated because of that?
Now that's a tough one. I don't know.
I'm happy.
That one?
Yeah.
Go ahead.
Yeah. So as one of the people up here, I want to thank you for coming here. And the natural selection is, in science, it's the fundamental basis of biology. That's science. Creationism is religion. Both have a place, but they shouldn't be confused.
So it's another polite way of saying we probably don't have an answer for you, but we wish we did. Thank you. Okay. Any other questions? Yes, a microphone here, please.
My name is Yichong. I'm a graduate student majored at electrical engineering. I have 2 questions. The first is, from your lecture, we know that some bacteria may know that the number of bacteria around them. So is it possible that we just release too much of this kind of signal protein, so the bacteria may think that they are already too crowded, there are too many of them, so they may grow slower or even stop growing. And my second question is that from electrical engineering, we have noise signal. So do bacteria have noise signal, so they may transfer wrong information? Thank you.
Those are great questions, and I'm going to answer them both the same way, with the same, so those are fantastic questions. So the bacteria, it's a little circuit, right? And you're right, so you guys get this question, if there's noise, like if somebody made too much of the signal or somebody made too little, how do you count correctly, right? And is there noise, right, so that they make a mistake? And so the reason these are called autoinducers, so a part of the system that I didn't tell you, because I had little simple slides, right, is that what happens is the bacteria are making and releasing the molecule's building up. And if an individual in the community detects enough molecule that it says, oh, I should do quorum sensing, the first gene it turns on is the gene that makes the enzyme that makes the molecule. So if I flip my bit, I flood the system with molecules, so these guys come with me, right? So it imposes synchrony and it filters out noise. right? And so it's not like it turns on like this. It turns on like this, right? And so that's why they're called auto-inducers, because I auto-induce the whole community, right? And it's a way to make sure that any noise in the system gets erased, because there's like this exponential rise in signal level, right? And so that's how, like, so they've counted to a certain number, and it doesn't matter if there's more of you. It matters the low threshold, and then everybody does the group behavior.
I would like everyone to give a round of applause for Bonnie and Daniel and Marty. Same time next year. But now let's repair to the outside and have some refreshments. Thank you very much.